Was it funny?
Hey, hurry. Everyone know when you go watch a video. Hurry, can you tell me what it says?
Did you watch that video, Caroline? We kept saying, hey, Max, because he was like watching it live.
I was like,
It's not like we were trying to ignore you.
I was trying to have a conversation with Max.
Okay, so remember you have this PowerPoint too. So this is a summary of the differences again. You should also know the similarities. Remember that there are 3 events that happen in meiosis that are completely unique. So the tetrads forming up, that synapsis, remember, is just the process of them connecting. The crossing over between the non-sister chromatids, that increases genetic variation. And at the metaphase plate, the tetrads line up. That's the second way genetic variation is increased. And then anaphase 1, the tetrads separate. So the homologous chromosomes are separating and not the sister chromatids like meiosis. So remember, genetic variation that happens in meiosis is what happens in the red bullet down here. It's a reshuffling of the deck of cards you already have. So the alleles already exist. You're just putting them in a new order. Okay? Brand new genetic variation, like new alleles happen because of random mutations. So we're not really even talking about that right now. What we're talking about, this genetic variation that is created during meiosis, is just a reorganization of alleles that already exist. Does that make sense? So it's important to know sort of the difference between new alleles and then just reorganizing the ones that are already existing. So there are, like I said, the 3 ways that genetic variation occurs is crossing over, which we cannot quantify. We don't know how much it happens, so you can't say it makes this many different combinations. We don't know. You can quantify independent assortment, which we talked about yesterday. That's 2 to the nth power. So I can make 2 to the nth types of eggs, and then someone else can make, like Fletcher, 2 to the nth types of, sperm. And so you can also, like, 2 people can have a huge number of different offspring possibilities. You take 2 to the n times 2 to the n. And that's how many different children, offspring, are possible between any 2 partners.
That's crazy.
It's a huge number, yeah. 3 to the 23rd is over 8 million already. So over 8 million times over 8 million is like insane. So that's how many different, I have the number in here somewhere, 70 trillion.
Oh, yeah.
Different offspring that are possible, right? Now, some people might say, well, why, do I still kind of look like my sister? Even though there's over 70 trillion different combinations, why do I still kind of look like her? Well, then that has to do with what we call linked genes, which we're going to talk about in chapter 15. But those are genes on the same chromosome that have a tendency to travel together. In a little while, we're going to do a meiosis lab. And I have these little chromosomes on here. And these little boxes indicate the genes, and the letters are the alleles. So genes on the same chromosome are said to be linked. They have a tendency to travel together through meiosis. Because when you line them up on the metaphase plate here, like all the genes on this blue one, this long blue one here that hasn't experienced crossing over, they're all going to travel together when those chromosomes split, the sister chromatids, right? The farther away, so let's look here on this one that has experienced crossing over, the farther away a gene is from the centromere, the more likely it is to be experiencing crossing over event. And it's because the farther you away are, the more like tangly it can get. Anyway, so genes that are linked and also close to the centromere usually will travel and they won't be separated. So linked genes can contribute to the fact that, you might look like your sibling, but also dominance versus recessiveness. And that can also, maybe you both have dominant alleles for a particular trait. Okay. So the third way genetic variation is increased through meiosis is actually after meiosis. It's called random fertilization. Or we would say increase through sexual reproduction. Okay, so you need to know those 3 ways that sexual reproduction increases genetic variation. 2 of them happen within meiosis, and the third one happens after it's happened. It has to do with a random sperm from daddy fertilizing random egg from mommy. Does that make sense? So it's random which egg is released every month, and it's random which sperm actually gets to be inserting into that egg. tons of sperm are released in every like ejaculation, but only one will be the winner.
That's what I always tell people. Like, never feel like you're not important because you are. You beat out so many other sperms.
Oh, that's right.
To get to where we are now. We are the sperm that made it.
Yes. Everyone has value. Yeah. So girls are also made of the sperm too.
No, I'm saying everyone.
Like, everyone. Okay, yeah. Sure.
Only half.
I didn't say that.
So I put up some pictures of like celebrities with their siblings.
Oh, that's Emma.
Oh
my God, Emma Watson. Like Emma Watson.
I know, especially with that short hair. Like,
That's Gwyneth Paltrow.
some of them look more similar than others.
Zac Efron's brother.
Anything like
that.
Yeah. I thought that was
Taylor Swift.
Yeah, I didn't see it.
Yes, that one.
They both look what?
No, this one.
How was Sean?
That's the one. I love my own.
Is that
They
the
probably wrote it. Yes, y'all didn't watch.
Okay.
Alright, and then.
Wait, you were.
Alright.
His brother's like young though, right?
Hopefully he'll grow it.
No, Zac Efron. Is his brother's name Ian?
Oh, that is what Zac Efron looks like in
Is it? Okay.
Zac Efron looks better. He looks like that weird little secretary face guy. Y'all. His face wasn't as chubby.
Yeah. Who's just gonna admire? I
You need
mean,
to go watch the work. Okay.
I know, right? Gosh.
I know, that's why I'm saying it.
That's a lot of
I love them. Okay. A couple of years ago, and one of my students was obsessed with the Giants.
Okay, Kevin, everybody hated Kevin because of the size. He needs to get rid of him.
I don't think they're that.
I'm into them now because he's connected the beard. Before it was just sideburns. It was bad.
They're just not.
Nick
I don't know.
Girly, show Kevin Nick. Yes.
Anyway, okay.
it's about time.
So the last thing I want to mention before we talk about nondisjunction and chromosomal abnormalities is that not all organisms use like the diploid haploid stages in the way that we are familiar with. So in the middle here with the frog, most animals do just what we do. So you have gametes that are produced through meiosis. The gametes will fuse, and through a fertilization event, produce your zygote, which will develop into a multicellular organism through mitosis. That organism itself makes the gametes through meiosis, and the cycle repeats itself. So plants are weird, kind of, because they do something called alternation of generations. They're actually kind of cool. But plants, like the big plant you think of, like a fern, you ever ferns during the summer, or even like a big tree or something, that big plant is something called a sporophyte. That's just what we call it, it's a sporophyte. And they actually produce haploid cells that are spores. And these spores will be distributed. And on a fern, the spores actually will land somewhere on a forest floor where it's really wet, and it will grow through mitosis to make a multicellular little plant. The entire thing is haploid. It's kind of crazy. Big trees and stuff don't do that anymore. But and you can move it wherever you want.
Listen,
I have all these tiles. I'm sorry.
can I put it back here?
Yeah.
Or are you gonna trip over it?
No.
All right.
I'll remember it's there. Just put it down. I'll probably just hang out over here. Thank you. And then this thing is called a gametophyte, and it undergoes meiosis to make, well, it just produces gametes through mitosis actually. And then the gametes will get together and form a zygote, and then blah blah. Anyways, we'll talk about plants next semester, but just know they're kind of weird. And then some algae, most of their life, like most algae, is actually all haploid. Most of the life cycle of an algae is a haploid organism, and they will undergo these little haploid guys will undergo mitosis. Why? It's like a Triscuit. I was like, that does not feel like a thing. Triscuit. Oh, you eat Triscuit? Not really. My fat cell.
I do not eat Triscuit.
Anyway, it's just different, so that's it. Okay, let me move on. Oh, next. Okay, so our next PowerPoint is about problems in meiosis slash chromosomal abnormalities. So, chromosomal abnormalities can happen for a couple of reasons. One, you experience something like we did in our cancer lab, where a piece of a chromosome breaks off and reattaches to like somewhere completely else in your genome. We'll talk about here. Let me actually just skip forward to those right now. Okay, so what we talked about in our cancer lab was translocation, the one on the very bottom. Okay, and that's where pieces of chromosomes, like whole chromosomes, whole pieces move from one area to another in the genome. Why this is bad? This isn't just one gene. This piece of a chromosome could contain hundreds of genes, and moving that whole thing somewhere else is pretty disastrous. Other things can happen on a chromosome structure itself. And so a part of a chromosome could be deleted. Maybe it got tangled up and it broke, and that when it formed back together, that piece broke off, which so you could have a deletion and deleted just multiple genes in there. A duplication, it happens actually really commonly that parts of genes and chromosomes could be duplicated, which could be a problem because now you have like too many of a gene. You don't just have one of them, now you have two or three. Inversion is when a piece breaks off and then it's stuck back in, but backwards, like the wrong way. And that's bad too, because when something goes in, it will read it the wrong way, not even produce a functional protein. So these are chromosomal abnormalities that just happen randomly, usually during different stages of, it could happen during mitosis, which would produce the cancers that we talked about in your body cells. If it happens in meiosis, the daughter cells or the gametes are going to have, all of them will have the problem, and we'll give it to the egg or the sperm, and then it'd be pretty disastrous because it would form every cell of the new organism would have this same problem. All right, other problems. Yeah.
So like, people who have like a problem in meiosis, that like when those happen, do they have like a higher chance of that happening in mitosis?
So no, if they were formed because their sperm had a chromosome that one of these is, like every cell in their body is going to have that same problem. So it's not like they're more likely for it to happen again.
So like, say they have translocation in meiosis, are they like at a high risk of having translocation in mitosis? Or is it like no?
No, there's no.
Where would the problem like happen in meiosis? Would it be like in crossing over, like
Yeah, so the bottom inversions and translocations are probably during like phase 1. And then up at the top, the deletions and duplications, that usually happens during replication due to random problems. That's when it would happen, like every. Okay. Now, having a wrong total number of chromosomes is a condition called aneuploidy. Do I have one here? He took my head. So aneuploidy is having a wrong number of chromosomes or incorrect. I'll spell it for you. It's having too many or too few chromosomes. Of maybe just one or two of them. Okay, it's not having too many or too few of all your chromosomes. That would be something, if you have like an extra of all of them, it's called polyploidy. And that's not this. But, and aneuploidy will occur through something called nondisjunction. And nondisjunction is when the chromosomes don't separate properly during either anaphase 1 or anaphase 2 of meiosis. Well, aneuploidy is that you have the wrong number of chromosomes. So if an individual is aneuploid, they have, and like, they don't have the normal number. They don't have 46, they might have 47 or 45. Or they would have 48 or 49. It just depends. I'll give you some examples. We already talked about that. Okay, so it can happen in two different ways. So first of all, it can happen during anaphase 1 of meiosis 1. So remember in meiosis 1, you have the tetrads line up at the metaphase plate. And these are supposed to separate, right? So that each daughter cell in this particular cell would have 3 chromosomes each, 3 duplicated for this first stage. But it might not separate properly. And if it does, like one cell would get all of the tetrad versus the other one would get none. It could also happen during anaphase 2.
No.
You just turn it on or has it been on? Shaq's like saying,
I can't hear this one.
oh, it's like flashing at me.
No, I have a question.
Is it on? It's on. Jack, it was on. It was. Yeah, it was.
It turned out, it's okay.
I probably
You're just, it's funny. You're just like looking at me normally like, that thing's flashing. What were you going to say, Alyssa? Were you going to ask something?
No, it was me.
Sorry.
So how would that show up like physically?
Well, let me show you. Hold on.
Oh, okay.
Hold on. It could also happen during meiosis 2 where the sister chromatids don't separate. Okay, so this is normal. Like I said, each was supposed to have 3, and then during meiosis 2, the sister separates, so each germ or gamete would have 3 unduplicated chromosomes. What should have happened in the cell? If nondisjunction occurs, like I said, if the tetrads fail to separate properly, so this one does not separate. So one of the daughter cells will have 4, 1, 2, 3, 4, and the other one will just have 2. And when these separate during meiosis 2, and then 4. So this is an egg, and it got fertilized by a normal sperm that has 3. Depending on this organism, that zygote would have how many chromosomes? 3 plus 2 is 5, whereas it was supposed to have 6. Okay, so that's aneuploid and it's missing a chromosome. And if this was the egg that got fertilized by the normal sperm, this would have 7 chromosomes. Now I'll give you a few minute examples of what happened. Hold on. So this is a picture in your book. I think this is in chapter 15. I don't think it was in 13, was it? Yeah. Anyway, so this is showing the nondisjunction event in meiosis 1. If the nondisjunction event happens in meiosis 1, all of the resulting gametes will be abnormal, all of them. Because the 2 resulting cells at the end of meiosis 1 are already abnormal. And so half of them are what we call N plus 1. So this happened on one of the tetrads. So they'll have N number of chromosomes plus the extra. And then half of them will have N number of chromosomes minus 1. If it only happens to one sister chromatid in one cell in meiosis 2, which is what this is showing, then half the gametes will still be normal, because half are normally over here, and then the other half will be abnormal. See that? Yeah. Questions about nondisjunction? So the result of this is that the baby, the resulting zygote, and the resulting organism will have the wrong chromosome number. They'll be aneuploid. So a trisomy is having 3 copies of a particular chromosome. And here we have sort of the example I was given earlier. That's our haploid number of N, normal, and then this is our N plus 1 egg. And when you fertilize, then you have trisomy, 2N plus 1, that's this extra chromosome of this one down here. A monosomy is when you're missing a chromosome, so you only have 1 copy of a chromosome. So in this case, our egg only had 2 instead of 3 chromosomes, and our sperm had 3, so our haploid here and N minus 1. So our resulting zygote will be a monosomy, 2N, and this one will only have just 1 copy. See that? So human chromosomal disorders actually occur really frequently in humans. A lot of times, you won't even know because the embryo is spontaneously aborted. That's called a miscarriage. And miscarriages happen quite often. Maybe your mom had one at some point in her life. I've had 3 or 4 friends that have had miscarriages. One of them had multiple miscarriages. My mom had a miscarriage. And usually it's because there was something really wrong with the embryo and it could not finish developing. And so it just, your body, it dies and you spontaneously abort it through a miscarriage. And it's really hard to go through that. I never went through it, but my best friend Elizabeth, the one I showed you a picture of yesterday, she did twice. And, you know, I know that it was probably for the best, like there was probably something wrong and it was going to happen anyway. So you know what I mean? You can't tell that to someone who's had one. You can't be like, well, so, you know, it's just really hard to deal with. Anyway, But it's because there's just too many problems. Yeah. No, there's actually other reasons. So when I got pregnant with the twins, I had to do fertility stuff. Like I give myself hormone shots, and then it made me release multiple eggs, which is why I actually had 3 fertilized eggs at the beginning. One just didn't finish developing, and I don't know why. They don't know why, but they made me take progesterone after I did that to maintain the lining of my uterus, because sometimes you have a miscarriage and it has nothing to do with the baby, but it has to do with the fact that some women, your progesterone level is because that corpus luteum thing, that yellow thing, I don't know if you remember the picture, but it stays in you and it produces progesterone and it maintains the lining of your uterus. The reason why you menstruate when you don't have a fertilized egg is because your progesterone levels fall and then the lining of your uterus slips off. So some women have miscarriages because for some reason they don't maintain their progesterone level even though they have a fertilized egg. And so they slip off the lining, and then there goes the baby. Does that make sense? So not all miscarriages are caused by that. Some, like the progesterone thing, are caused because of other problems. So some women, like, they gave me progesterone just in case. They were like, you're not going to go through all this, spend all this money, and then lose your babies because you can't even maintain your lining. So anyways, usually it's because you have multiple copies of different chromosomes. And let me pause for a second and show you a karyotype. So this is obviously an abnormal karyotype. And we'll talk about this disorder in a minute. But just, I want to, the 4 and 5 are here. Ignore that for now. But all these are the normal number up here. And humans will not tolerate. So usually if you have an extra chromosome 1, that's when the baby will probably be spontaneously aborted. For some reason, the chromosomes that are the longest have the most genes on them, obviously. And they carry the most information. And we talked about the dosage thing yesterday? Yeah. So you need 2 copies of the proteins that are made here normally, right? And then 2 of all these other ones. And if you have an extra number 1, you have times 3 of all of those proteins. And it usually creates such a biochemical problem because you have just too much of those chemicals, the embryo just can't finish developing properly. Or if you're just missing 1, you only have 1 instead of times 2, you just have times 1 of it. You don't have enough of those chemicals to allow normal development, and the baby will die. So it happens, like I said, early on. Most miscarriages happen within the first trimester. They usually don't happen later after that. That's why a lot of women don't say to people that they're pregnant until they got to 16 weeks or so, because they passed that sort of danger zone of the miscarriage happening. There's a teacher that used to work here, and his wife got pregnant, and he told everybody when she was 6 weeks pregnant, which was really early on, and she ended up having a miscarriage from 10 or 12 weeks. And I remember and he didn't tell anybody she had a miscarriage, and I was like, how's Kelly doing, blah, blah, and he was like, oh, shit, and I was like, oh. So I never told anybody I was pregnant until I was 16, 17. A few of my students actually, yes. But,
I predict you're about 17 weeks.
They guess because for sure.
no, well, with the twins, I have to get correctly.
Miss Healthy was social security.
Oh, I wasn't mean. I ate all the time. I just anyways, okay. So some conditions are tolerated because the balance is not as disturbed, basically. The ones that are more survivable are the smaller chromosomes, the ones that don't have as many genes, which is why trisomy 21 is Down syndrome, and that's actually not uncommon. But the really small chromosomes. So you can have 3 copies and it's not such a problem. And also the sex chromosomes are much more tolerated. Having extra copies of the X is much more tolerated. So Down syndrome is one of the examples, and like I said, it's trisomy 21. You can see the 3rd chromosome right there. And it's one of the smallest ones, and so it's not such a big deal. But having that 3 times 3 of those proteins still creates such issues that every single person that has Down syndrome, they all have a certain set of symptoms, which is why we call it a syndrome. If you think about it, everybody that you know, think of a picture of someone that has Down syndrome, they all kind of look the same. They're shorter in stature, Their face is kind of round, moon-shaped. They have almond-shaped eyes. Like they're not skinny. I mean, they're not fat. They're kind of like stout. You know what I'm saying? They all look like that. And it's because having that third set of the chromosome does that. They all have heart issues. And most of the time, people with Down syndrome don't have an extended lifespan because of other issues they have. They'll die a little bit early on, not super early. They will have a range of mental ability. Some are super high functioning, some not so much. It really just depends. Some can live independently, some will never be able to live independently. The frequency of Down syndrome, and here I'm going to come back to what we were talking about earlier, correlates usually with the age of the mom. And if we look at this graph over here, this says incidence of Down syndrome per 1,000 live births, and down here says age of the mother. So notice it's really low until around the age of like 33. It starts to rise, and then it really rises after age 40. When a woman is 40, it's 1 in 105 pregnancies. Down here at the age of 49, 1 in 12. The reason that happens, and really this would be the incidence of any chromosomal abnormality, like there's other ones. There's trisomy 13. There's all the other sex chromosome problems. It has to do with this fact that the women, you're born with all the eggs that you have, and they're stuck in meiosis 1. So the spindle apparatus is all stuck. And the older those eggs are, the more problems there are with the functioning of that spindle apparatus, the older you are. And so it's much more likely that there'll be problems. They won't connect to the superchromatids or to the tetrads, and then they won't separate properly. Then you have more or 2 Does that make sense? Yeah.
So what is chromosome 21 in charge of?
We could find the genes for it. I don't really know. I used to have a chart, like a big poster, but it's got old and gross. I can look.
So if the mother is like an okay, like the dad is like really old, that would be so much.
No. Yeah, because men they continuously make new sperm forever.
Yeah, we stay first.
Yeah. But also that's the reason why we go through menopause. It's to help protect this from happening. Because after, 49, 50, it's like it would go really high. So menopause is a protective sort of function to prevent you from releasing any more eggs that could have a lot of problems with their spindle apparatus. Does that make sense? Because they're just too old at that point. And that's why we go through menopause. And then the rest of your age just stay there. So, that's Down syndrome. Like I said, there's other types. We'll get to the sex ones later, but there's trisomy 13 that creates a certain set of symptoms. Anyway, so there's genetic testing that women can choose to have done if they want to. There's 2 types. You can do an amniocentesis, which is what they're showing here, where they stick in a hypodermic syringe into the amniotic fluid and take out some of the fluid. In that fluid, they will do 2 things. They will find fetal cells and karyotype them and get a picture of the baby's chromosome. They also will analyze the fluid itself for the presence or absence of particular proteins that could indicate another disorder, a genetic disorder the baby could have, not necessarily a chromosomal disorder, like a genetic disorder, like a mutation in just one gene. So that's what they will do, and to see if there's a problem. So did I tell y'all the story of, y'all know Crawford Latham, the sophomore? I have her karyotype. I'll have to show it to you. She has an older sister named Grace. And when I first started teaching, Grace was a sophomore in one of my classes. And Grace's parents divorced. They both remarried, and her mom got pregnant again when Grace was in high school with Crawford. And so when we were talking about this stuff, she's like, oh my gosh, I'm going to bring you my mom's karyotype. She had all this done because her mom was much older when she got pregnant with Crawford. And so, like I said, if you're older, they will highly recommend you have this done because you're at a much higher risk for developing chromosomal problems. Like I actually have Crawford's karyotype. It's really strange. Can you imagine if I came up to her last year and I was like, you're Crawford, I thought your half sister Grace and I have your karyotype. She's like, what? Like I gave her a copy of it. That's what it looks like. This was from the USC School of Medicine that they printed out and like gave to Crawford's mom. Like this is Crawford's chromosomes and she was normal. It says on here somewhere normal XX, 46XX. And this is kind of what the somatic, like smearing looks like, and then the computer organized it. Anyway.
That first one.
It's kind of crazy, right? Anyway.
What's the happen? Oh wait, you're right. You're right.
So,
How much does it cost to get
oh, I don't know. I never had one done.
About 5,000.
They told, that's how the story went. Yeah. Yeah.
But the moms that really have like Down syndrome, is that more like
Not at all. Not at all. Yeah. Not at all. One of my friends from high school, Sarah, her sister has like 5 kids. One of, I think her second child has Down syndrome, but the rest of her 3 did. She wasn't old when she had all her kids in her 20s. So, yeah, but no, it's sort of like, no, not at all. Random.
You're gonna tell the story.
Oh, yeah. So when I got pregnant with the twins, I wasn't old. I was 30. I wasn't that old. But they told us, they give you all these booklets, like you can do all this testing, you know, whatever, to see if your child has different disorders. And at first, Fletcher was like, you know, his whole idea of like us having a kid was we were going to have one kid. He's the only child. We were only going to have one. And like, if the child had a problem, we were going to find out and like prepare, get everything ready, and like be super prepared. And then when we got pregnant, and the doctor was like, there's at least 3 in there. Like I told you the story. It was like, you imagine like doing the ultrasound and like, oh, there's the baby, and like it's a sweet moment. He's like holding my hand at first, and the doctor's like, oh, look at that, you won the lottery. There's at least 3. He like lets go of my hand and sits down, and I'm like, okay. It's awesome, right? Anyway, but they tell you, you know, you can do all this stuff, and I kind of wanted to do that too, but once I was pregnant and I'm like asked these questions, I'm like, well, if I did this testing and something came back abnormal, would we know which one it is? They were like, not necessarily, no. Because if you do the biochemical stuff of the fluid, you don't know what baby it's coming from necessarily. And so we were like, well, do we want to know? And we're like, no, I don't want to know. Like, I was like, it doesn't matter if something's wrong with one or both of them. We'll just deal with it when they're born. And like, I just don't want to know. So we didn't do it because it didn't matter to us. But we never thought we would feel that way. It was really interesting. Yeah.
have like Down syndrome?
If it came from the same egg, like if they're identical. So yeah. Fraternal, it's not probable. Yeah. Yeah.
I've got mine in my closet.
Yeah. So I'll have to bring him and show you my twins. I've got a DVD of them. It's crazy.
I think my mom.
Yeah. I don't ever look at them. But they can detect other abnormalities doing the ultrasound. When you're around 21 weeks, 17 to 21, they'll do a big ultrasound and measure length of the baby's bones, the circumference of their head, all kinds of stuff to determine if they're developing normally. Sometimes they can detect Down syndrome by doing that stuff. A lot of times a baby with Down syndrome has an abnormal vessel cord. Normally there's like 2 arteries in a vein, or 2 veins in an artery, 3 vessel cords. But if a baby has like a 4 vessel cord or a 2 vessel cord, that could indicate a big problem in the umbilical cord going. But they can detect those things and say, you know, and suggest other testing anyway. So they can also determine problems just by doing the ultrasound, which they'll do anyway. So that's that. So, like I said, the sex chromosomes are much more tolerant in terms of problems with abnormalities. And there are a bunch of different syndromes, and I'll talk about it. So Klinefelter's is having any number of X's other than just 1 and a Y. So it's XXY, it could be XXXY, 5 X's and a Y, it doesn't really matter, but as long as there's just 1 Y and more than 1 X, you have Klinefelter's. There's something called trisomy X in females. I'm going to talk about all these. There's Jacobs syndrome, which is X and then more than 1 Y. Could be 3 or 4 Ys, not just 2. And then Turner syndrome is the only human monosomy that is tolerated. There is no other human monosomy. But monosomy X is Turner syndrome, and that is survivable. The rest of them would not be survivable.
Zero.
X nothing, like no other X and no Y. So Klinefelter syndrome, like I said, is having more than 1 X and a Y. 1 in every 2,000 live births. We say live because you don't really quantify the ones that are spontaneously aborted anyways. They have male sex organs, so they will have, you know, testes and all that, but they're sterile, so that means they don't make gametes. And usually there's problems in that spindle apparatus because they have the extra X's and it just won't finish. So they won't be able to reproduce. They kind of look a little more feminine because they have the extra X's, and they'll look kind of curvy, more like a girl and not just like a guy, and they'll kind of just look soft. Does that make sense? They might have breast development, and they won't produce facial hair like a guy would. And they'll be tall, like not abnormally tall, but just tall, not short. Normal intelligence. And so here's an example karyotype. So they would say 47 XXY. So they have 47 total chromosomes and the 2 X's here.
So it is true that like estrogen makes you taller. Like you get taller when you get estrogen.
No, not necessarily.
Oh.
I'm just saying they're not abnormally short.
Um,
Women in general are shorter than men.
well,
I mean, I'm not, obviously, but
I was about to say you got me.
got my dad. Okay.
Okay, can you explain this to me? How come I'm short and my dad's really tall, but my mom's short too?
The
Why don't I
genes
just get the
for
middle?
height. Well, that's a good question, and it has. Hold on. It has to do with the genes, but the genes for height are not sex linked at all. Not that I know of.
Yeah.
It's also polygenic. Okay, so we're not there yet. The polygenic means that, think about all the different heights a person can be. It will make like a bell curve, right? There can be super tall people and super short people, like everybody in between. It's much more common there's something in between, right? Hair color the same way. Human intelligence the same way. Eye color the same way. Anything that looks like a bell curve and you take all the phenotypes out is determined by multiple genes. It means polygenic. So it's really actually much more complicated. The genes for height are probably all over our genome, and there's definitely more than 2. There's a lot of them. Most human characteristics are actually polygenic. So it's complicated, is my answer. Human skin color, there's a bell curve. It's multiple genes. Anyway,
I have a 5'4"
talk about that next
body,
later.
6 foot,
Yeah.
This boy, he's like 4'11", and his dad is 5'2", and his mom is like 5 if that.
Yeah.
Yeah, he's really.
He just has a lot of receptors.
Y'all the same.
So here's a 4 X's and a Y.
There's more X's like
Not really. Doesn't make it any worse. It probably happened because it depends. There's a worksheet we're going to do later, but so like a tetrad could have not separated properly. Like so all 4 X's, like in the mom, could have gone to one cell, and the whole thing might not have separated in meiosis 2. So the egg could have had 4 X's in it. Does that make sense?
so that wasn't what happened.
Not necessarily, because he could have got an X and a Y from the dad. It really just depends if the egg and the sperm were both messed up or just one. You know what I mean? It really depends. Sort of hard to tell. Okay, Jacobs syndrome is having at least more than 1 Y and 1 X. These are all males, 1 in 1,000, so it's a little more common. They're a little bit taller than normal, and they're more active than normal. They're more testosterone, whatever. Normal intelligence, so they can have some learning disabilities, such as just some issues learning. Your learning disability doesn't really have anything to do with how intelligent you are, your IQ. Delayed emotional maturity, meaning that it might take them longer to just, just to be able to handle stuff. You know, guys in general mature later than females. So this would be just delayed even more. Normal sexual development. So not necessarily. Trisomy X is having 3 X's. This is a female, and what's really interesting is that you won't even know if you have it unless you have a karyotype done. These are healthy females that will produce gametes, and everything is fine, like totally fine. So the reason why is something actually really cool. Did we talk about the calico cats? We already did this. Yeah. So it's because of the bar body. So just their 2 extra X's are turned off. They just turn them all off. So it doesn't really matter. They still have 1 functioning X, and they're totally fine. Turner syndrome, like I said, is the only monosomy X. The monosomy that's tolerated is called monosomy X. These are females. They are sterile. It's more rare, 1 every 5,000. they have a set of symptoms. So their neck is what we call webbed. You can kind of see it on this little girl right here. It kind of goes out. They're much shorter than normal. And I mean, this picture up here is actually really helpful. They can have a lot of these things. They're sterile, so they won't make any eggs. They might not even menstruate. There's a girl I went to high school, actually all through school, that had Turner syndrome, and no one really knew what was different about her, why she was different, and just that she was different until I took a bio. And I remember seeing something like this, and I was like, oh my gosh. Anyway.
Okay, so what about like the animal of that working out?
It wouldn't be like that. This is really different. Like, I've never seen.
I wonder if it was different after a certain
Like that? What's up?
Okay, like if they always turn off this extra X's, then why does it matter if they only have one? You know what I'm saying? Like if it's trisomy X and they just turn off the other two, then why does it matter? If there's only one. If they only use all.
What do you mean? Why does it matter? Why do we care?
Why does it like impact them? I guess like if they only have one.
It doesn't impact them, which is why it's okay.
For Turner.
Yeah. Oh no. Oh no.
Like
that's a really good question.
Like,
the second X is not turned off until a certain point in development.
okay.
And so the other one is probably used. Does that
So
make
it's like
sense?
a developmental.
Yeah. Yeah. That's a good question. Sorry, I wasn't. Yeah.
So monosomy Y is the thing?
No, and that's a good question too. The reason why monosomy Y isn't a thing is because, well, we'll get to it in a little. Look, I was looking at your stuff. The reason why monosomy Y is not a thing, I mean, it could be a thing, but that would cause development to stop and that fetus to be miscarried. And the reason is because the X chromosome, you can see how much longer it is compared to the Y, but these are just certain disorders. But the X chromosome carries a ton of genes on it that have nothing to do with sex development or gender at all. It has like a number of normal stuff. You can have hemophilia. Hemophilia is a blood clotting disorder, and you produce a lot of clotting factors by genes that are located on your X chromosome. So there's a lot of like normal developmental stuff that you need that's found on your X. If you don't have it, you won't survive. Does that make sense? The Y doesn't have really anything except for like the male determining factor in this. And so you can live without a Y because it just makes someone a male. That's all. Okay. And yay, we're done.
Can you look up what like chromosome 21 represents, like what it contains or whatever?
Yep. Oh, see, this is like the picture I had. Those are disorders.
Breast cancer.
Yeah, I used to have a poster of all the different disorders that were attributed to the different chromosomes, and like I said, I got old. This is from that. I mean, there's so many, but yeah.
Only familiar where that's on the title.
This was a cartoon. Oh, never mind. Anyways, I don't know. This is my X and Y. Sorry. Here you go. We could look it up later. I don't really know. A lot of stuff. Okay, so we're finished with our notes. Let me stop this. I want us to work on our lab. So I want you to work with one partner in this lab. Now I have copies, but I feel like copies are okay in this lab. So if you don't want to use copies, you can just use the second part of your lab to do the first part of your lab too. So I have up here 2 color sheets of paper, and if you look at them, y'all can see one has lowercase letters and one has capital letters, right? This is indicating from like mommy or from daddy or vice versa, it doesn't matter. Okay, so you're going to use a cell that has 4 chromosomes, 2 pink ones and 2 blue ones. Now, the reason there are 2 copies on here is because there's a duplicate for each one. So you're going to have to model. So in part 1, here's the lab. Sorry, Billy and I, but no one else does. Who didn't pick one of these up? Some of you did.
What does it pick up taxes? We did not read.
On the board.
Here's that. There's so many buttons.
Oh, yeah? Yeah, mine are like that. No, there's some in the drawer. I have some really cool there texture.
My mom literally had a bag of my hair
You have one?
for my first hair.
I'm not sure if I old I've got a live video of the ultrasound and a 4D one, so you can see their Okay, so for part 1, you're going to simulate it, and I have on here these copies, but you can just cut these out and use these to simulate it if you want to. It might make you a little bit not crazy. Instead of doing a video, I want you to actually show it to me. So when you're ready, studying the class, show it to me. Otherwise, you can show it to me tomorrow. I don't really care what you, well, let's try to show me all that before you leave today. And then fill all this out. And then the next part of the lab is where you actually have to use these chromosomes here. And you have to simulate crossing over. And so anyway, just read the directions. It tells you what to do. If you're confused, please ask. Some people in the class are super confused. But if you read, you won't be that confused, and then there are questions on the back. You don't have to finish this for homework. Your homework is actually to do chapter 14 notes, because once we finish this lab tomorrow, I'll give you probably the first half of class tomorrow to finish it. We're moving right into chapter 14. That's when we'll get stuff done before Friday. You might not be here Friday. So,
Sitting right here.
okay. Yeah, so work with one partner. You need 1 blue and 1 pink sheet and scissors. And let me know if you need me. I'll be up here doing letters of recommendation for something and looking at the sheet of notes. Go back here.
We all do this.
It's not like there's a problem.
Maybe now.
I'll do it tomorrow. Your letter, it's weird because a lot of people, I was
Probably not. That sounds like a
they're breathing or not. Yeah, but it's weird. Like, look, this is all the other people. I've gotten emails from CollegeNet, and I don't know why. Here, let me try.